Assalamualaikum and salam sejahtera. My name is Siti Sarah Marzuki. Today, I will present my scientific case study entitled Engineering a Plastic-Free Future for Food Security, Biotechnology Strategies to Combat Microplastic Contamination in Blood Cockles, Anadara granosa from Kuala, Selangor, Malaysia. Imagine eating seafood that looks completely fresh but already contains invisible plastic particles. Microplastics are plastic particles smaller than 5 millimeters and are now widely found in marine ecosystems worldwide. Blood cockles, Anadara granosa, are one of Malaysia's most important aquaculture species and provide an affordable source of protein for many communities. However, because blood cockles are filter feeders, they continuously filter large volumes of seawater, making them highly vulnerable to accumulating microplastics. This pollution threatens not only marine ecosystem but also food safety, public health, and Malaysia's food security. For this study, I selected Kuala Selangor, one of Malaysia's major blood cockles aquaculture hubs. According to a recent study published in Marine Environmental Research 2024, all sampled blood cockles contain microplastics. The dominant microplastic detected were fibers, fragments, and films with particle size ranging from 0.05 to 0.1 millimeters. More importantly, Kuala Selangor recorded a polymer hazard index or PHI of level of 5, indicating a very large contamination risk. The student also estimate a dietary intake of approximately 0.029 micrograms per kilogram or body weight per day, demonstrating that human exposure through seafood consumption has already begun. Microplastic first enter blood cockles through filter feeding in contaminated seawater. They are then transported from the gills into the digestive gland and other tissues where they accumulate. This accumulation triggers the excessive production of reactive oxygen species or ROS, leading to oxidative stress. As a result, blood cockles experience DNA damage, inflammation, apoptosis, and cellular toxicity. Over time, these physiological effects reduce growth, weaken immune responses, and impair reproduction. Finally, when contaminated blood cockles are consumed by humans, these microplastics can enter the human food chain and potentially affect human health. One promising biotechnology solution is CRISPR-enhanced PETase technology. PETase is an enzyme originally discovered in the bacterium Ideonella saccharensis, which is capable of degrading PET plastic. PETase first breaks PET plastic into an intermediate compound called MHET, which is then further degraded by MHETase into terephthalic acids or TPA and ethylene glycol or EG. Through CRISPR-Cas9 gene editing, scientists can modify the PETase gene to improve enzyme efficiency, stability, and degradation rate. CRISPRs contain benefit and risk. The benefits include reducing persistent plastic pollution, protecting seafood safety, and supporting sustainable aquaculture. However, there are possible risks such as horizontal gene transfer, unintended ecological effects, and public concerns regarding genetically engineered microorganisms. In summary, microplastic contamination is no longer just an environmental issue. It has become a serious challenge to food security, aquaculture, sustainability, and public health. Protecting our oceans means protecting our food, our health, and our future. Thank you for your attention. I hope this presentation has shown how science and responsible innovation can work together to build a plastic-free future for food security.